supposed to be taught to you, so that should be clear to you. If I'm training somebody who is purely in science and wants to utilize the flow cytometry, I would be scaling it slightly higher than what is going to be given now. And in any case, the didactics require that by the end of the lecture, there should be some learning outcome. So what we are going to cover today is not wholesome of what needs to be covered in flow cytometry. Flow cytometry can at times become a bit more complex. It used to get more complex to me when I was training, when I was doing my postdoctoral training. So, and I'll just mention to you the types of flow cytometry which are there. And interestingly, even having known the basics and concepts of the flow cytometry, I came to discover about some aspect of flow cytometry called cell sorting, that you can separate the cells and put in different test tubes, go back to the laboratory where you are doing your experiments with the cells of your interest, and then discard the ones which you are not interested in, what is called flow activated cell sorting. So in brief, what we are going to go today is just basics of flow cytometry. And a student doing training in medicine and BCHB should focus much more on the clinical application of the flow cytometry, and also the limitation of flow cytometry in respect to other molecular techniques, which I discussed with you only last week or the other week, the PCR. I mean, I would have been much more interested not to have the topic PCR alone, because there are other techniques which I'm teaching other group of students, particularly those who are doing medical laboratory science and technology, expanding beyond the flow cyto, beyond the PCR, to enlighten them on other techniques which you can use in detection of pathogens. For instance, the herpes viruses, which are commonly associated with the malignancies in the tropics, the Epstein-Barr virus, the Kaposi sarcoma-associated herpes virus. And these technologies came recently, so they are much more important for you also to know them as you near completion of your medical training. But of course, you're still in the medical school. We are just in 4th year, so I think you still have 3 more years. Okay. Now, for the case of the flow cytometry, just like the name suggests, there's something which is detected in the fluidic system. So then cells which last time we were talking about, you extract the DNA, then amplify a segment of it. In this case here, we are going to look at the property of that cell, and the cell is subjected to some laser light, and then excitation of that cell is going to emit signal. So that signal is what is captured, hence the flow cytometry. And the outline or the teaching outcome, by the end of this lecture, you should be able to know the principle of the flow cytometry and some working terms, definitions of terms which are going to come. And basically, I've mentioned to you what can be measured by flow cytometry. So where would this flow cytometry be important to you as somebody who is doing a medical course? I mentioned to you that that cell which you need to measure must be suspended in some fluid. So therefore, we'll be looking at what are the components of the flow cytometry: the fluidic component, the optics component, which means that laser light being subjected to the cell of your target, and then there is capture of the signals which are released, hence the electronics. Now, I wanted to change something. Something here a little, if you pardon me. Then I put it this way, and then I put it this way. That looks more good. You'll be happy with it. And why do I do that? To make you get a distinction on the components of the flow cytometry, that it has got the fluidic component, the optics component, that laser light which is subjected, and then the electronic component, which means the signal processing from the excited light. Then for you to get this signaling, there must be what is called the fluorochromes, which means the dyes which are used to label these cells or the markers on the surface of this cell. But of course, the detection of the target molecule can also be the DNA, which is inside the cell. So it is not only that these dyes are going to mark the surface of the cell properties, but can also detect for you the DNA and also the chromosome number, which is within the nucleus. Then you go to that analysis where you have got what you have the flow cytometry files, or you go to analysis you checking at what is the population of these cells which are spread much more onto the left, which are these cells which are spread much more to the right. We'll see all that when we'll be talking about the scattering of cells. Hence, the term gating. Gating simply means you divide the whatever, it's not whatever, you divide the square region of the population of these cells, and the 4 quadrants, you'll be having the upper outer quadrant, upper inner quadrant, left quadrant, and you have also the bottom left, right. So you'll see the population of these cells, or if the chromosomes which you are looking for, are they spread towards the left? But if they're spread towards the left, is it upper left or the down left? Right, is it the upper right or the down right? That is what is called the gating. Then you can do the statistics and analyze the population of cells you're dealing with, or the number of chromosomes you're dealing with, what proportion of them are scattered towards the right, what proportion of them are scattered upwards. But we'll see all that. So the basic principle of flow cytometry is just measurement of light which is scattered by particles. So these particles could be molecules which are expressed on the surface of the cell, or you can talk about DNA in the chromosome, and that definitely would be captured in the nucleus. And the fluorescence observed when these particles are passed in a stream through a laser beam. Hence, I'm just taking you back to these components of the flow cytometry. We get the fluidics, we get the optics, which is that laser light, and then the electronics, which means now the signal processing. So when we talk about the definition of terms in the flow cytometry, and I've mentioned to you the flow cytometry becomes study of cells as they move in the fluidic suspension. So basically, even if you had got a cell culture, or you can do also a touch prep at your level right now, you are advanced in pathology. So you want to make analysis of these cells, whether they are tumor cells or what, and from a biopsy, you can just touching preparation on a slide, touching that fresh tissue which has come from the theater onto slide to collect the cells which you are interested in. Then you suspend that cell into fluid buffer, which would enable you to have single cell suspension. So that cell, if you pass it through now the laser, it will get some excitation, and that excitation can pick now signal which you can analyze the properties of that cell, whether it is tumorous or not tumorous. So you're learning today a very nice thing. And I mentioned to you, I need to tell you about flow cytometer. And up to the time I was doing my postdoctoral training, I had not gotten exposed to what here is called the flow activated cell sorting. We have the cell sorters, which is numbered here 3 in the bullets here. But the types of flow cytometers which are available, they are traditional flow cytometer, they're acoustic focusing cytometers. The acoustic focusing cytometer is just typically that traditional flow cytometer, but there is addition use of ultrasonic waves instead of the laser waves. Then you have got the cell sorters, which I have indicated also down here, FACS, which means flow activated cell sorting. And this one implies fluorescence activated cell are separated from those ones which could not be activated. And what it does is very good. I was very impressed. You want to separate a population of cells. For instance, you students by now have got little knowledge, if not much, that, for instance, a cell infected by Kaposi sarcoma-associated herpes virus, not only that, cell infected by Epstein virus leads to a number of tumors, including non-Hodgkin's lymphomas and also primary fusion lymphoma for the case of the Kaposi sarcoma-associated herpes virus. And the trick of these viruses is that they undergo latency program. At what point they hide within the cell, and that is how they cause the pathogenesis. So at an ample time, they trigger what is called lytic program from the latency program. So the lytic program ensure that the cell now divides very quickly, and some become even immortalized, which means they divide continuously. And in that way, therefore, you can look at, if you are studying these cells, you collect the population of cells which have undergone lytic phase from those ones which are still in latency phase. What will help you do this is what is called this flow activated cell sorting. Very important. And looking at the technical aspects, by time you'll be rotating in immunology laboratory and get, I used to prepare itinerary program on what you need to cover, but I think I've not looked at it this year. I think Dr. Baraza has prepared it. I will ask if she has included the itinerary for the technical aspect of the flow cytometry. So when you come to flow cytometry, and for those who may have access also to the CCC down there, for those who may have access to CAVI up to there, for those who may, no, we don't have flow cytometry work in Unity, but a number of places in the university there have got the flow cytometry, which if you get time, you don't need necessarily to wait until the time you rotate in immunology to learn about it. So then sample preparation becomes the first primary point on the protocol or the procedure of the flow cytometry. And why is it important? I've mentioned to you, before these cells are ran in the fluidic channel, which you'll see, these cells must be put in a single cell suspension. Like I gave you example of a touch prep. You can take a tissue of, say, a suspected Kaposi sarcoma. You can get a tissue of suspected case of maybe a lymphoma, and then you do a touch prep, harvest the cells, and then these cells are suspended in a single cell suspension fluid, BAF, of course. And after that, these cells are incubated in test tubes and or microtiter plates. By now, you know ELISA plates, and so microtiter plates is simply ELISA plate. And these case cells with unlabeled, or you can have them labeled with fluorescent, incubated with fluorescently conjugated antibodies. And then later you can now analyze the cells in the flow cytometry. So this is very key, important for you when somebody asks you about steps in the flow cytometry procedure. This slide looks to me too busy, but it's not as busy as one slide, which is quite much ahead. Don't worry about it. I've tried to make it much more less busier. The second step in the flow cytometry, once you now prepare those cells in the microtiter plates or the test tube, you now need to stain them with antibody. Why? Because these antibodies are the ones which are going to detect for you the markers on the surface of this cell, or these antibodies are the ones which are going to detect for you the copy DNA number in the chromosome within the nucleus. So once the sample is prepared, you have these cells rocked. The best term is rocked, shaking it, okay, and then incubating with the antibody, which are coated with fluorochrome conjugated antibody. And they must be specific to the marker which you're looking at. If I'm slow, you tell me, because you can just type there so I can adjust my speed. So these markers which you are targeting, be it in the surface of the cell or in the nucleus, you can now incubate them with fluorochrome conjugated antibodies. And after that, you have these cells washed. They're washed to remove those ones which didn't bind to the antibody. And then you run them in the flow cytometry, which will now excite these cells according to different wavelengths of light, subjecting them to laser light. And from there, you can move now to the analysis stage to check the proportion of cells which are scattered in the direction which you would be interested in. We'll see that kind of scattering in a short while. So the conjugation using these markers can either be done using the direct pathway, not direct, but direct mode or indirect mode. You remember, I don't know, it's me who taught you about indirect ELISA and then the direct ELISA. When you are talking about, Dr. Ndungu, I'm in class, I'll call you. When you're talking about indirect staining, excuse me, students, When you are talking about indirect staining, these cells are incubated with an antibody directly conjugated to a fluorophore. But when you are talking about indirect, the fluorophore conjugated secondary antibody is the one which is now labeled, then the primary antibody is not conjugated. So that is not quite much to disturb your head. I made it a stressing point for you that in your learning, focus much more into where will I utilize this flow cytometry in my medical practice. Then when you are interested in doing masters, then we can negotiate what to add to you, filtering these details further. And when you reach doing your PhD, I scale it further up, so you become much more excited, like this cell which we are going to use as examples to be excited to release signals. And I mentioned to you the intracellular staining procedure allows for direct measurement of antigens present inside the cell. So it's not only the cell surface where you can check these markers of the cell, but you can also detect these markers in the cytoplasm or the nucleus of the cell. Then that means you have to cause little friction on these cells to expose the antigens which are in the cytoplasm or in the nucleus. Then after that, you now run your sample. And running sample, you must have control beside it. Just like you're doing any other procedure, you have to have negative and positive control to guide you onto the detection of the target marker which you are looking for in your cell. And the flow rates in the cytometer are set, and then sample will run in the fluidic medium. I'll show you just that shortly. But before we go to that, let's look at what measurements can be made in the flow cytometry. I'm interested in the granularity of the cell. I'm interested in the size of the cell. I may be interested just in markers on the surface of the cell, like somebody doing the flow cytometry classification for leukemia, different types of leukemia. And you know that the leukemias originate from these turbans of the leukocytes. And these leukocytes with the white blood cells would have markers on their surface. So if you talk about, for instance, leukemia, which has got predominant expression, B-cell leukemia or T-cell leukemia. If I'm talking about B-cell leukemia, then definitely I'll be looking at markers which are expressed on the surface of B-cell. If I'm looking at a T-cell leukemia, I'll be using that fluorescent T-labeled antibody to mark for me markers on the surface of the T-cell. So the fluorescence which is done will be helping to bind to the target molecule on the surface of the cell. And you can also have addition of calcium ions, sensitive dyes within the cells, and also fluorescent proteins which are expressed by the cells. And you also have the DNA dyes which can be able to mark for you the DNA within the nucleus of the cell and not necessarily on the surface of the cell. So a lot of things would you be looking for here, maybe granularity of the cell, the size of the cell, or markers on the surface of the cell, like cluster differentiation, CD4, we have CD19 for the B cells, CD20 for the B cells, CD5 for the B cells. You get also CD7 and all that. So these are markers which are expressed on the surface of the cell when the cell is differentiating through different stages. And when I mentioned to you the scattering mode, there's something I've not shown you here. So you have got forward light scatter when you are analyzing your cells. Cells would more or less be scattered forwardly, or cells can be scattered sidewardly. And this is what is called forward scatter cells, or you have got the side scatter of these cells. But then all this is geared towards maybe I'm measuring the size of the cell against granularity of the cell. You get me? Or I'm using the maybe I want to check how the immune system has responded to TB in maybe in a latent TB patient. So let me check maybe interferon gamma expression against, say, the interleukin 6. So I will be scattering the interferon gamma maybe on the side scatter against interleukin 6 on the forward scatter. How does that look like? It looks like this. it looks like this. The side scatter here and tells you the proportion of the cells which would be distributed. Like I mentioned to you, cells could be scattered on left, lower, or I mean, you put your hands and look at this square diagram between you, and you can be at the center and look at the right side of the square or the left side of the square. The right side of the square still would be having upper right and the lower up, just like you have on the left side would be having lower left and the upper left. So when I'm talking about the forward scatter is that scattering of the cells towards the horizontal. Then side scattering of these cells towards vertical. So look at this first circular cell. You have got the smallest and least granular population of cells would have the least scatter compared to the more granular cells, and now this bigger cycle. So you have got larger and most granular cells will be scattered much further. And here, therefore, if you want to measure the proportion of cells in your disease condition, you could be having population of granulocytes scattered on the forward scatter, then see at the quadrant where they are, look compared with the lymphocytes, which are less granular, they are less scattered and remain down here at the forward scatter, yes, and also representing side scatter. And this helps me now to interpret the proportion of my cells. In between there are those monocytes, which are in the intermediate between the granularity of the lymphocytes and also the granulocytes. So this is how I can mark the population of cells if I don't do I mean, this is not routinely done. So don't think like you can substitute this kind of technique with the full blood count to help you mark the monocytes, granulocytes, and all that. But this can complement, especially now when you have got advancing from the full blood count, you've checked hematological parameters, and then now you look at this patient I'm dealing with could be having some kind of, say, leukemia. What are the proportion of cells which can help me now make a diagnosis of the entity of leukemia I'm dealing with? And I mentioned to you all the time you have to have a negative and positive control. So if you do a fluorescent isothiocyanate intensity, I'll come back to this fluorescent isothiocyanate Fe and phycoerythrin. Don't be bothered about them right now. But these are dyes which are used to mark these cells. So each time what I'm indicating here is that you must have a control, negative and positive control when you are run, which will help you understand that the test has run well. So like this case, what we are seeing, when you'd be wanting to have this side scatter and the forward scatter, for instance, you can use a different dye which is helping you to show the side scatter and a different dye which is helping you to show the forward scatter. Here, the forward scatter, which is the dye which is most commonly used for the forward scatter, is the fluorescent isothiocyanate, and the side scatter is the phycoerythrin. You'll see this. I've got a tabular list of all these dyes which you can use. But the more or less is the same. If you divide these quadrants, you do not have to divide it proportionately because the population of these cells will be scattered differently. I mentioned to you, like, you have got this bottom left. Here are those cells which may be less granular or having less of the marker which you are targeting. Those which are having more of the marker which you are targeting could be scattered either in this larger quadrant or the far right, this green quadrant. And the major components of a flow cytometer, if I may now bring you back, I mentioned to you that the flow cytometer constitutes fluidic system, optical system, and then electronic system. The fluidic system is this where now the cells are passing. You remember I mentioned to you, you prepared that your cell into suspension, and then now channel it into a flow channel. And there is excitation of laser light, which is subjected to these lights as they pass through the tube. And what has passed down here is therefore just a waste which would be discarded. But in all this, then at what point there is excitation of these cells in different wavelengths. And this is how the optical system would be enabling you to detect these cells. And then at the electronic system, you'll amplify that signal and then channel it to computer for further interpretation. This is how you'll be interpreting it here, therefore later, the signals of the cells which were subjected to that laser challenge. So the components of a flow cytometer, I mentioned to you, you have got sample intake port, which we've seen here. You have got sheath and the waste reservoir, which you've seen down here. No, you see down here, the waste to waste container. And then you also have the flow cell, which you are seeing the cells from a sample. Then you have got the laser. I've shown you here excitation laser, which is challenging the cell. Then you have got the optical filters, which you have there, the optical system filters according to the wavelengths of the light. Then you have got the photo multiplier tubes or photodiodes, and then the signal processor, the detection system, as it occurs in, say, the computer. And this is what it is. So you have got the cytometer fluidics, create the laminar flow. You have got the flow cell, the extreme end. Our important area is the sample stream, the sample with this cell suspended in the solution or the fluid. And then the sheath is just to give a protection for this channel, which the cells will be passing. Challenge these cells each time with a laser beam light, and these cells will get excitation, which can now release for you the signal. And this is what is happening. Yes, you see, this cartoon is looking at it very well. So each time these cells pass, they are challenged with the laser light, and they will be remitting the signals. And then the ones which are required would be proceeding to the analysis. Those which are not required will go to waste. Here is where I mentioned to you the third or fourth class of the flow cytometer, the cell sorting. So in this case, cells are sorted into the test tube. Those I mentioned to you gave you example of the pathogenesis of the Kaposi-associated herpes virus or Epstein virus, which goes into latency. But then at the lytic stage, they can now transform that cell into lytic phase. So lytic phase means that cell now divide and proliferate and making becoming much more tumorous. So in this case, you are interested in only collecting those cells which have turned out lytic and leave alone those ones which are not turned lytic and still remain latency. Hence, you are sorting these cells according to the population of cells which you are interested in. And once you collect this tube of cells, you can now go back to the laboratory and culture them and expand them and do experiments which you need with them. If I give you typical two-color cytometer configuration, which I don't want to go much into detail, like I mentioned to you, you're not doing a PhD here. And these cells are excited at different wavelengths, either 560 nanometer short pass dichromic mirror, which is helping to capture the signal in the photo microtubes 2, or you have got the side scatter photo microtube 1 up there. And one thing which is quite important, you can have this analysis of cells at different beams of light. So you can have, say, 488 nanometer laser beam, or you can have these cells analyzed at a different nanometer laser beam like 560. This will help you to evaluate the proportion of cells which are excited at different wavelengths of light. You must be sure that for every technique, just even as you're doing the hematoxylin use in staining, or you're doing maybe the immunohistochemistry technique, or even you're doing the PCR technique, there must be some element of background interference. And this background interference can come from the flow cytometry autofluorescence of your cells. The cells can also autofluoresce. And therefore, there must be correction measure which is instituted to ensure that this autofluorescence is not giving you wrong results, which you can sign out or institute management of the patient based on this autofluorescence. So the background noise, we call it noise. The background noise must be limited. So where does this background noise come from? I've mentioned autofluorescence, or you have non-specifically bound antibodies and also free antibody in the sample serum. When you are measuring cells, you pick cells from blood, you would also be thinking like part of the cells could, the serum contained also antibodies which could react or could fluoresce and give you signal which are incorrect. But I'll show you in some last slide, how to correct this, this noise or background signal which you don't want. And the level of autofluorescence I mentioned varies with the wavelength of excitation. It can be much more in the higher wavelength, like you're seeing here at 560, compared to lower wavelength, like the 588 nanometer. So we have got the highest excitation of this autofluorescence occurring in the signal of fluorescent isothiocyanate, and followed with the phycoerythrin detectors, and it is far much lower in the APC, which I'll give you the naming all in a table which I've shown back. These are dyes which are used to label the antibodies before they detect for you the target molecule, be it the granularity of that cell, be it markers on the surface of that cell. But I've given you a table of all these names of the dyes which you can use. And then you are interested in measuring the detection efficiency, like I mentioned to you, when you are doing the flow cytometry, you have to take cognizance that some noise in the background or fluorescent signals will be there. So you need to be analyzing the detection efficiency and also the background, which is non-specific signal, and you don't need it at all. We didn't need to go into detail on this, but you know just to take it that when there is background noise or interference, you have to be cognizant that you have to compensate before you interpret your results. Oh, it is now here. So the common fluorophores for antibody conjugation or labeling, you have got fluorescent isothiocyanate, is called isothiocyanate. This person must have been poor in English. It is fluorescent isothiocyanate, not fluorescent isothiocyanate. It is fluorescent isothiocyanate. And you have also others. Alexa Flow. Well, I liked Alexa Flow so much when I was doing my advanced training in the flow cytometry, but it was the first time I was using Alexa Flow channel, so it was exciting to me. And I've mentioned you can also use the phycoerythrin. You can use phycoerythrin texared or phycoerythrin cyanine, iridium chloride protein, which is indicated plus P, and all this. So depending on what you are interested in detecting, and depending on the type of molecule you are interested in and type of excitation laser you are using in the flow cytometer and approximate emission peak which you are interested in, you can use different of these fluorophore dyes in conjugating your antibody. And this is what is mentioned here. For instance, you have got signal which would be peak at 530 nanometer for the 488 Alexa portion. So these dyes which you are using for the conjugation of antibody before it detects to you your target molecule will be dependent on what you are targeting. What molecule do you want to detect? Type of excitation laser which you are using in that cytometer, and what is your expectation peak which you are needed. So you cannot use an excitation peak of say more than 600 when your molecule is known to excite at 530. Again, I think I need to, this I will correlate with you probably if I'll be teaching you the clonality testing. How do I distinguish between tumor and a non-tumor, which is technique of clonality testing? Then it will come out to you quite clear. These signals and the peaks at the wavelengths which you are interested in. You cannot analyze a molecule which is exciting at 300 when definitely it is documented should be analyzing the peak at 530. This will come out clear to you when perhaps I'll be teaching you about clonality testing in lymphoproliferative disorders. And so you have got the mission of fluorescent isothiocyanate against the phycoerythrin channel, and this could be easily analyzed from the inception of the molecule, like this fluorescent isothiocyanate or the phycoerythrin. If you have got phycoerythrin known to be exciting at 585 nanometer or fluorochrome 1, PTC, is exciting at 530, you cannot interchangeably mark your antibody with PE when you are targeting to analyze a molecule which is exciting at 508 at 30. PE is known to show excitation at 585 and above. And I told you when you have got the spillover or you have got that noise or unnecessary signal, here I give you a typical example. You have got PTC mean fluorescence, and you also have got PE mean fluorescence. And for the uncompensated mode, which means it still has that noise, you have got the negative signal and also the positive signal. And when it is already compensated, of course, the negative signal will still remain the same, but the positive signal will just shift now slightly. Same thing will occur with the phycoerythrin. Well, there, interestingly enough, the phycoerythrin, you have got the negative signal can change as well as the positive signal. To get the percentage spillover of this, you typically take the positive signal of the PE minus the negative signal of the PE on the compensated. Then divide with the positive signal on the FITC minus the positive signal in the FITC minus negative signal in the FITC, again on the compensated. You don't want to go into uncompensated because I mentioned here, we are compensating for the noise which has been realized, and you are trying to balance if it has been able to give you the correct position of the cell. So you have got how do you pronunciate this? Hierarchical gating, and this gating just means trying to mark the population of your cells in terms of the distribution. And I mentioned to you something which I need to bring you to. Look at these squares, and this is how you analyze the flow cytometry. So I need you to look at the square on the top right, top right, where you're analyzing interferon gamma through the FITC and against interleukin-2. And I mentioned to you, sometimes you're not only thinking about the markers on the surface of a cell, but maybe you want to analyze how the body system responded to maybe a TB bacteria. And TB is, as an intracellular pathogen, is known to induce the cytokine immune response. So I would be analyzing, say, the interferon gamma, which are released in this immune response against the TB antigen. And when I mark the side scatter, the forward scatter with phycoerythrin, the cells which are the proportion of molecules. Here we are not talking about the cell. Proportion of these molecules which are distributed in the proportion of these molecules which are distributed in the left in the forward scatter or right in the forward scatter. Then I come analyzing them down in the side scatter or up in the forward scatter. So these proportions P6, P7, or they're just helping you to regroup these cells, and that is what is called gating of these cells. So I start from analysis of these cells. This is probably a donor, and I want to analyze the proportion of molecules in this blood of the donor. So I have got the forward scatter and the side scatter. So from there, I analyze it further by saying, let me analyze the CD4 and CD8 proportion of these cells. Then I come analyze the CD4 marked with a different dye against the CD8 marked with a different dye. I analyze the proportion of the cytokines response in this donor. Then I can go ahead until I get details of these cells which I wanted to characterize. So this is what flow cytometry helps you analyze. And this is the slide I was mentioning to you, is so much busy, and so I want to go slowly on it, but I don't want to go slowly because you say that I'm slow. In the application and uses of flow cytometry, this is quite important for you. Flow cytometry is used in several fields, which include molecular biology, pathology, immunology, virology. You are not doing plant, but plant biology or marine biology. Some of them are common applications to both, which will include in pathology, for instance, or in disease state. Flow cytometry is used in clinical labs for the detection of malignancy in body fluids. And I've mentioned to you typical examples in leukemias. Cytometers like the cell sorters I've mentioned to you can be used to separate cells. For instance, you want to check pathogenesis of the lymphoma cells during infection, which are infected by Kaposi's associated herpes virus. So you can sort those cells which have reached the proportion of lytic stage against those ones which are still in the latency stage. I've mentioned also that fluorescent flow cytometry can also be used to detect the content of DNA in the chromosome, and by using fluorescent markers which are labeling or targeting or can detect this DNA within the chromosome. And to do that, I mentioned to you, you must rupture these cells a little without breaking it overtly. in what is called rocking, just shaking on the rocker. And with that one, then it will cause a rupture on the surface of the cell to expose the chromosome. So flow cytometers also allow the analysis of replication of cells, and this you can do by using dye for 4 different stages of cell. You know, the stages of a cell cycle. You have got the G1, then we go to any clever student. From G1 you go to which phase of cell cycle? This is the problem of online teaching. It's like I'm talking to myself. You go to now the M phase, then you go to the S phase, you go to you go to M phase, you go to G2, then you go to the S phase, and then the cell cycle continues like that. You must have been taught about this. And I mentioned to you also the acoustic flow cytometer, which is just the traditional cytometer, but uses a different laser compared to the flow cytometer, which are used to study multi-drug resistant bacteria. And this is quite important for studying why there could be drug in a store, maybe approved for supply by KEMSA, but you as a doctor, you are treating this bacteria and you're not seeing anything working. So you'd apply this kind of study to analyze the multi-drug resistance in bacteria using acoustic flow cytometers. And the different stages of cell cycle or cell death, apoptosis, necrosis, can also be detected by flow cytometer based on the differences in the morphology and biochemical changes. For instance, if you get the apoptosis and cells undergoing apoptosis, you have got the Ki67. An anti-apoptotic marker is the BCL2. So you can use also that to check the proportion of cells which are undergoing apoptosis. You can check proportion of cells which are undergoing the necrosis, or you can check the proportion of cells which are continuously proliferating, undergoing mitosis, like the marker, the Ki67. So look at how you have a lot of things which you can do with the flow cytometer in the medical practice. How about the limitations of the flow cytometer? The process doesn't provide information on the intracellular location or distribution of proteins, so that is quite a limitation of it. And the flow cytometer, just like PCR, which we talked last time, is expensive and needs skills. I don't know where I put that on point number 4. It is expensive process and requires highly skilled qualified technicians. And again, point number 2, over time, the debris which are aggregated might result in false results. But I've told you how to compensate this when you get problem with that. And also the treatment associated with sample preparation is time consuming and really can delay the turnaround time if you are relying on it to make second opinion in the management of the patient. So with that, I think I've cleared you on the flow cytometer. Apart from these references which I've given you, I think I need to add you other references which you can pick when you'd be interested in doing your personal studies. Any question? Any place you want me to revisit and clarify? This is the problem with online teaching. You don't know whether your students are still there or they are left.
Oh, we're here, Doc. Thank you for the class.
You know this online teaching. I'm teaching students who have not even seen their faces. Is that not bad?
You'll interact with us soon.
Sure.
Yeah.
Anyway, the online learning is good because it also saves me time. But sometimes I don't think it's good because maybe somebody's cooking in the kitchen and just put their connection and it's not there.
No, we're here, Doc.
Like I'm seeing one there in the kitchen. He's called Lavin Ogada. Ogada, are you there? Oh, he's there.
I am here.
Oh, I thought you were in the kitchen. Anyway, so if you don't have any question, because again, these lecture notes, sometimes the way they come, you may not absorb it immediately. But then once you get the lecture notes, go through it slowly. You have my email. If you have any question, just post. Actually, I'm not here. I'm not getting this part. May I get clarification? I'll be able to respond to you through email. On the squares, we have got fit C and PE. So are we using two antibodies on the same flow of cells? Yes, you can use even up to 4. You can use even up to 4 because what I've indicated to you there is only 2 properties of a cell I'm looking at. Maybe I want to check the like these cells, how much interferon gamma are they releasing? How much interleukin 2 are they releasing? So in that case, I'm comparing interleukin 2 against the interferon gamma. Then I can come later be looking at a different property of these cells. Maybe now I'm looking at what proportion of these cells express CD4, what proportion of them are expressing the CD8. So at each point, you will be having two fluorescent labeled antibodies to detect to you because you are comparing something against something. That is when we talk about the forward scattering and the side scattering. So at each point, at each single time, you have to use 2 dyes to help you scatter these cells forward and scatter these cells sideways. Any other question? Okay, if you don't have any more questions, then you can have a good afternoon. Class rep remind me by around 4 or 5 there to send
Recording stopped.
And the other ones.
Yes, Doctor, I will.
Okay.
I also wanted to ask, these flow cytometry and all these immunological tests, are there supposed to be some practicals in the clerkship during pathology?
Yes. When you